Shall we start? Shall we start? Okay. So, very good evening to all. Today we are going to discuss some important questions in physiology with regard to the upcoming UHAS exams. Okay. These questions I have selected. They are most commonly repeated in different papers. So, let's start with the first question. First question is, define GFR, mention its normal value and describe the factors which are regulating it. Okay, it's a very important question. It's asked in the recent paper also. Okay, so let's start with it. So, what is glomerular filtration rate of GFR? It's the amount of plasma ultrafiltrate formed in each node. That means it is a rate, i.e. it is measured in terms of time. So, we know that the different filtering units in the kidney or the functional unit of the kidney is the nephron. And there are about 1.2 million nephrons in each kidney. And the glomerular filtration rate means that all the nephrons, that is in both kidneys, all the nephrons are functioning and they are filtering and they are making a filtrate. And all nephrons in both kidneys, which are filtrating in 1 minute. That's what that whole thing is called as glomerular filtration rate. Okay, it can be measured in indirect experiment and animals also in humans by measuring the plasma level of the substance and the amount of substance that is excreted. I.e., this filtrate is derived from the blood plasma. We know that the blood is passing to the kidneys in the glomerulus via the glomerular capillaries. Okay, and Bowman's capsule is the part of the nephron that is doing the filtration. And in the urine formation, there are basically 3 steps, and the first step is the glomerular filtration. Firstly, we filtrate the amount of certain things from the plasma, and after that, there will be a tubular reabsorption. Certain substances which are filtered from the plasma, they are reabsorbed because they are more potential substance that we are needing for our normal body maintenance. And hence, they are reabsorbed from the tubules. Okay. And certain substances which are to be excreted, they can be secreted from different parts of the tubules. That is the part of the nephron which are doing the secretion and reabsorption. Okay. So filtration is taking place in the Bowman's capsule, and the secretion and reabsorption of certain substances, they are taking place in the tubules. Okay, that's a general overview of the urine formation. So, filtration is the first step in the urine formation. So, what is the amount of GF? What's the GFR? The normal GFR in adults is about 125 ml per minute. Or when a day is concerned, it is about 180 liters are filtered from our both kidneys during a whole day or in 24 hours. Okay, that's the glomerular filtration rate. So now coming to the different factors which are affecting the GFR. Okay, first factor and the foremost factor is the renal blood flow. For obvious reasons, we know that when the renal blood flow is increased, that means the amount of plasma that is traversing through the glomerular capillaries will be more, thereby increasing the filtration. So, when the renal blood flow is more, what happens to the filtrate? It will increase. That is the most important factor that is necessary for the glomerular filtration is the adequate and ample blood flow to the renal system. So, it is directly proportional to the renal blood flow. And normally, the normal blood flow to both kidneys is about 1300 milliliter per minute. And usually the renal blood flow, it is controlled by auto-regulation. Okay, it's not depending on many things, but it is auto-regulated. By the kidney itself, it is controlled. Okay, different mechanisms are there for the control or the auto-regulation of the renal blood flow. Second factor is the colloidal osmotic pressure. In lower classes, we have studied that when the osmotic pressure is increased, water is drawn more towards that side. That means what? When the colloidal osmotic pressure is high, that means the filtrate will be less, i.e. the amount of filtration will be lesser. For example, in dehydrated states, we know that the amount of water present in the plasma will be reduced and plasma proteins will be more. And hence, water is more drawn towards the blood side or the plasma side. Hence, the filtration will be less. And hence, colloidal osmotic pressure that is inversely proportional to the GFR. Okay. So, when the colloidal osmotic pressure or the protein content of the plasma is increased, GFR will be decreasing proportionately. Okay. Also, in hypoproteinemia, if the protein content is reduced, the water is more drawn towards the filtrate area. So, filtration will be more. Okay. Next is the tubuloglomerular feedback. This can asked as a short note. This can be asked as a short note also. So, this is very important. So, it is usually this glomerular filtration rate is regulated through a feedback from the renal tubule. It is called tubuloglomerular feedback. Just 1 second. Okay. So, the structure that is responsible for the tubuloglomerular feedback is macula densa. Okay. So, macula densa of the JG or the juxtaglomerular apparatus is responsible for the tubuloglomerular feedback. Okay. When the filtrate that is passing through the terminal portion of the thick ascending limb of loop of Henle, what happens is the macula densa act as a sensor. What is the thing that is sensed by the macula densa? It's the amount of sodium chloride. Okay. The amount of sodium chloride means the water content is also sensed. Along with sodium chloride, we are filtering the water also. Okay. Sodium chloride and water are proportionate. Okay. So amount of sodium chloride can determine the amount of water that is filtered. So it senses the concentration of the sodium chloride in the filtrate and accordingly it alters the blood flow and the GFR. When the sodium chloride level that is sensed by the macula densa is more, that means the filtrate is more. The filtration is taking place rapidly and somewhat unwantedly it is filtering from the plasma. And hence that is sensed by the macula densa. Thereby, it produces thromboxane A2. Okay. So thromboxane A2 means what? What is the function of thromboxane A2? So macula densa is producing thromboxane A2. If the sodium chloride that is detected is more, that means more water is filtered and hence it will produce thromboxane A2. It will cause the contraction of the JGS cells. That is leading to the constriction of the afferent arteriole. Afferent arteriole means it is bringing the blood from different parts of the body to the renal system. Okay. So constriction of the afferent arteriole means what's the effect of constriction of afferent arteriole? The renal blood flow is reduced, thereby reducing the filtrate. So the blood flow through the glomerulus is decreased and decreasing in GFR is happening. Conversely, if the sodium chloride is less, that means the filtration is not taking place appropriately and hence filtration has to be increased. For that, this macula densa is again producing prostaglandin E2 and bradykinin. This will cause the dilatation of the afferent arteriole, thereby increasing the blood flow to the glomerulus and the glomerular filtration rate. Okay. Also, efferent arteriole means the blood that is taking the efferent arteriole is taking the blood away from the renal system. And renin-angiotensin, renin is produced from the macula densa. It will cause the activation of angiotensin 2 to angiotensin 1 to 2. That is causing the constriction of the efferent arteriole. And this all actions, it will increase the blood flow to the glomerulus, thereby increasing the filtration rate. Okay. So this is very important. So the structure responsible is macula densa of the juxtaglomerular apparatus. What are the chemical mediators that are produced from the macula densa? First is for the constriction of blood vessels, it is producing the thromboxane A2. Prostaglandin E2 and bradykinin is produced for the dilatation of the afferent arteriole. Okay. And renin is produced for the constriction of the efferent arteriole. Okay. So GFR is mediated by tubuloglomerular feedback. Next thing is glomerular capillary pressure. So it is directly proportional to the, i.e. the GFR is directly proportional to the glomerular capillary pressure. Normal pressure is about 60 millimeter of mercury. And when it is increased, that is glomerular capillary pressure increases, GFR also is increasing. Okay. So it is also depending on the renal blood flow and arterial blood pressure. Next is the contraction of the glomerular mesangial cells. When the glomerular mesangial cells, they are situated in between the glomerular capillaries. So contraction of glomerular mesangial cells means reduced blood flow, thereby reducing the GFR. Next is the hydrostatic pressure inside the Bowman's capsule. Okay, this features you can remember hand in hand. That is the osmotic pressure and the hydrostatic. These are opposing factors. When the osmotic pressure is increased, hydrostatic pressure is dropping. Okay, so GFR is inversely proportional to the hydrostatic pressure in the Bowman's capsule. Bowman's capsule means inside, that is the amount of filtrate is more. That further, that decreases the amount of filtration. Okay, in the Bowman's capsule, not in the capillaries. When the Bowman's capsule, it is having more water, or the hydrostatic pressure inside the Bowman's capsule is more. That means the filtration is more and it has to be reduced. So GFR is inversely proportional to the hydrostatic pressure inside the Bowman's capsule. Okay. Usually, hydrostatic pressure in the Bowman's capsule is increasing in the conditions like obstruction of the urethra and edema of the kidney beneath the renal capsule. Next is, this all we have discussed already. These are all effects of certain renal blood flow. Okay. Constriction of the afferent arteriole. That is, constriction of the afferent arteriole means renal blood flow is reducing and thereby reducing the GFR. Next is constriction of efferent arteriole. That's a little tricky thing. First is, initially when the constriction of efferent arteriole, what happens is when the efferent arteriole, the blood coming from the renal system is increased, what happens is the amount of blood in the glomerular capillaries initially increase, thereby increasing the GFR. But if all the substances are already filtered and no further filtration is going to take place, efferent arteriolar constriction, that's the outflow is blocked. When the outflow is blocked, the inflow also reduces and thereby decreasing the GFR. That is, in initial case, what happens is when the filtration is in the initial stage, the constriction of efferent arteriole will increase the GFR, whereas in the final stage or after the filtration, if the efferent arteriole is constricted, what happens is the filtration will not be effective. Further blood will not be coming to the glomerulus, thereby decreasing the GFR. Next is the systemic arterial pressure. It's usually the arterial blood pressure is responsible for the blood supply to the kidneys also. When the systemic arterial pressure is high, what happens is filtration will be more. Also, some damage can also occur to the filtering apparatus. What is the filtering apparatus? It consists of three things. First is the endothelial cells. It is having certain gaps and there is glomerular basement membrane. Also the podocytes. Okay. There are also specialized cells which are aiding in the filtration. These three things constitute the filtering unit. Okay. So this may get damaged during hypertensive events. In hypertensive patients, we know that these plasma proteins are also filtered along because of the damage of this particular podocytes and different parts of the filtration units. So systemic arterial pressure is responsible for the flow of blood through various organs, including the kidneys, and hence what happens is when the pressure is increased, filtration will be more. Okay. But remember one thing, increase in the mean arterial pressure up to 180 millimeter or reduction up to 60, it will not alter the renal blood flow or GFR because autoregulatory controls are there for these things. Okay. And hence, this above 180, below 180 and so, okay, from the range of 60 to 180, there is no change in GFR because of the autoregulatory mechanisms. And if the variation is above 180, that is about a condition called malignant hypertension, or very high hypertension, that is above 180 millimeter of mercury is very high blood pressure. And below 60 is also low, the hypotension. Okay. So, beyond that, the GFR is changing accordingly. Okay. Because of the failure of the autoregulatory mechanisms. Next is sympathetic stimulation. Okay, usually we know that afferent and efferent arterioles, they are separated by the sympathetic nerves. We know that there are two components of the autonomic nervous system, sympathetic and the parasympathetic. Parasympathetic means it is firing when the person is normally resting or in a normal phase, normal situations. Only the nerves which are firing is a parasympathetic. Sympathetic nervous system is firing in emergencies. Okay. For in emergency situations, what happens is our BP shoots or raises. That is mediated by the sympathetic nervous system. The mild or moderate stimulation of the sympathetic nerves usually do not cause any significant change, because it's very important that it should not be under the control of any of this autonomic nervous system. It should be auto-regulated. I.e., we know that renal blood flow is auto-regulated, not under the control of sympathetic nerves alone. So mild or moderate stimulation of the sympathetic nerves, it do not have any significant change in the renal blood flow or the GFR. When strong sympathetic stimulation is occurring, what happens is severe constriction of the blood vessel, i.e. the constriction of the blood vessels all over the body is taking place under strong sympathetic stimulation. By releasing the neurotransmitters, i.e. noradrenaline or adrenaline is secreted in large amounts in the body during the strong sympathetic stimulation. In such cases, renal blood flow as well as the GFR should be altered. Okay. Then the autoregulatory mechanisms will be failing in such conditions, i.e. under strong sympathetic stimulation. So the effect is more severe on the efferent arterioles than on the afferent. I.e. blood flow as such is not increasing, but the efferent arterioles will be constricted, preventing the blood flow, preventing the taking of blood away from the kidneys. So initially there will be an increase in filtration. We have already studied that when the efferent arteriole is constricted, initially GFR increase, and after that, after a particular amount of time, what happens is the further constriction of the efferent arteriole will not be having any increase in GFR. Why? Because afferent arteriole is also affected. Blood is pooling there and no further blood is coming to the glomerulus. So, however, if the stimulation is continued for more than 30 minutes, there is a recovery for both renal blood flow as well as the GFR. Okay. So, in case of strong sympathetic stimulation, there is alteration of GFR. GFR will be somewhat more, okay, in the initial stage, but after 30 minutes, it is restored. Why this happens? We know that after the release of this sympathetic neurotransmitter, it will be fading. This effect is fading due to the reuptake mechanism and the degradation of the neurotransmitters, and hence after 30 minutes GFR will be restored as well as the renal blood flow is also restored. Next is the surface area of the capillary membrane. So GFR is directly proportional to the surface area of the capillary membrane. Okay. And if glomerular capillary membrane is affected in, say, some cases, if renal cell damage to the nephron occurs, then it will reduce the GFR because of the damage and reduction of the surface area of the capillary membrane. Also, the permeability of the capillary, if it is more permeable, that means more filtration will be occurring. If in some cases like hypoxia, lack of blood supply, presence of toxic agents, etc. will alter, i.e. it will increase due to the necrosis of the cells. It will increase the permeability of the capillary membrane, thereby raising the GFR. Okay. In such cases, when damage is occurring, even plasma, proteins, blood cells, everything is filtered and excreted in the urine. Okay. And the factors like hormonal factors are also affecting the GFR. So these are the factors that are increasing the GFR. Atrial natriuretic peptide. Natriuretic means something that is increasing the urine formation, and hence these are hormones which are responsible for the rise or increasing the GFR. Okay, atrial natriuretic. Natriuretic means something that is causing the increase of urine. That means these substances will be increasing the GFR. Okay, ANP, BNP, cyclic AMP, dopamine, endothelium-derived nitric oxide, that will cause the vasodilatation. Okay, prostaglandin, PGE2. This all increases the GFR, and decreasing things are angiotensin 2, endothenins, noradrenaline, platelet activating factor, platelet-derived growth factor, prostaglandin, F2. Okay. So these are the hormones which are affecting the GFR. Next question is define blood pressure and describe the baroreceptor reflex mechanism in the blood pressure regulation. This is a very important question. Chemoreceptors, baroreceptors, and the regulation of blood pressure as such can be asked. So, what is blood pressure? It's defined as the lateral pressure that is exerted by the column of blood on the wall of arteries. So, arterial blood pressure, it can be defined as the lateral pressure which is exerted by the column of blood on the wall of arteries. In simple terms, it's the pressure exerted when the blood flows through the arteries. Okay. Generally, the term blood pressure, that means arterial blood pressure. So there are different blood pressures like venous blood pressure, etc. are there. But in general terms, the term blood pressure simply means when blood pressure means arterial blood pressure. Okay. Now, what are the things that are regulating the arterial blood pressure? So, in normal, even under normal physiological conditions, BP varies. You have heard about the white coat hypertension. That is when the doctor is trying to check the BP of the hypertensive person. In normal conditions, due to some fear or physiological response, BP may be raised. So we have to measure BP after some 15 minutes and allowing the person to relax. And after that, taking the BP, then the BP will be somewhat lesser if the patient is under medications. Okay, so in normal physiological conditions also, there will be certain changes in BP that's occurring. However, immediately in such cases, immediately it is brought back to normal level in a normal person. Okay. That's due to the well-organized regulatory mechanisms in the body. So, our body has 4 regulatory mechanisms to maintain the blood pressure within normal limits. First is the nervous mechanism. These things can also be asked as short notes. Okay. Nervous mechanism or short-term regulatory mechanism. Second is the renal mechanism or the long-term regulatory mechanism. That includes the RAS system. Okay. Renin, angiotensin, aldosterone system, etc. is coming under the renal mechanism. Third is the hormonal mechanism. And fourth is the local mechanism. Okay, these are the 4 things which are generally regulating the arterial blood pressure. So, coming to the baroreceptor mechanism. Baroreceptors means what? The receptors which are sensing the pressure differences. Okay. There are chemoreceptors and baroreceptors which are controlling the blood pressure. Here we are going to learn in detail about the baroreceptor mechanism. Baroreceptor means something related to pressure. So, which gives response to change in blood pressure according to, pressure. And they are also called pressure receptors, pressure. And what is the situation or the location of the baroreceptors? They are situated in the carotid sinus as well as in the wall of aorta. What are the functions of baroreceptors? So, when blood pressure increases, it will sense the pressure difference and activates the mechanisms which will try to reduce the blood pressure. Okay. Baroreceptors are activated and it sends stimulatory impulses to the nucleus of tractus solitarius via the glossopharyngeal and vagus nerves. Okay. Glossopharyngeal and vagus nerves are responsible for the baroreceptor mechanism. And after reaching the stimulus is reaching, when the blood pressure is high, the baroreceptors are activated. That is sending impulses to the nucleus of tractus solitarius. And this nucleus of tractus solitarius, it will be acting on vasomotor center. And the vasomotor center has 2 components, a vasoconstrictor area as well as a vasodilator area. So in order to reduce the blood pressure, it has to inhibit the vasoconstrictory center and stimulate the vasodilatory mechanisms. Okay, so inhibition of the vasoconstrictor area, it reduces the vasomotor tone, and reduction in the vasomotor tone causes vasodilatation. And due to the vasodilatation, we know that peripheral resistance will be reduced. Okay. And simultaneous excitation of the vasodilator along with the inhibition of the vasoconstrictor area, that is reducing the TPR or the total peripheral resistance, it is activating the vasodilator center also. So that will increase the vagal tone. So vasodilator center is activated, that will increase the vagal tone, thereby somewhat suppressing the heart activity. Okay, so it will inhibit the heart. Vagal tone means increasing vagal tone means vagal tone will decrease the heart activity or inhibit the heart, and hence rate and force of contraction of the heart will be reduced, leading to reduction in the cardiac output. So decreasing peripheral resistance and reduction in the cardiac output will in turn try to reduce the BP. Okay, that will be leading to the BP to come back to normal. So What is the function of baroreceptors? When BP is increased, the pressure is sensed by the baroreceptors and it will be sending its impulses to the vagus nerve as well as the glossopharyngeal nerve. It will be acting on the vasomotor center, and in the vasomotor center, it particularly inhibits the vasoconstrictory center and stimulates the vasodilator zone. So, when the vasoconstrictor area is suppressed, total peripheral resistance will be reduced. Also, when the vasodilatory center is activated, that also decreases the total peripheral resistance. And vagal tone will inhibit the heart activity, thereby reducing the heart rate as well as the cardiac output. These all factors will bring down the blood pressure. Okay. So you have to learn the location and the function of baroreceptors. When the blood pressure is decreased, i.e. the blood pressure is dropping, then also this baroreceptor can sense the pressure difference, i.e. the pressure will be less, and it will cause the inactivation of the baroreceptors. When the baroreceptors are inactivated, there will not be any inhibition to the vasoconstrictor center. So vasoconstrictor center is no longer inactivated, so vasoconstriction can occur. It will lead to the increase in total peripheral resistance, and cardiac output will be increased, thereby trying to raise the blood pressure level. Okay. So, information regarding the blood pressure within a range of 50 to 200 millimeter, it reaches the vasomotor center via the carotid. There are two types of baroreceptors. One is the carotid and aortic baroreceptors. These are functioning in different situations. The carotid baroreceptors are functioning when the blood pressure is within the range of 50 to 200 millimeter of mercury. And if the blood pressure is ranges about 100 to 200 millimeter of mercury, the information is going via the aortic baroreceptors. And both the carotid and aortic baroreceptors, they are stimulated by the rising pressure than the steady pressure. When the blood pressure is rising, these are sensed by the carotid as well as the aortic baroreceptors. Okay, and usually their response is depending on this range of blood pressure variation. So, since this baroreceptors, they are acting in accordance with the pressure variations, they are called pressure buffer mechanism or system, and the nerves from the baroreceptors, they are called buffer nerves. Okay, because they are sensing the pressure difference and acting accordingly, they are called pressure buffer system, or simply this nerves which are supplying this baroreceptors, they are called buffer nerves. Now coming to another important portion. This is also repeated in different question papers. I've seen multiple times this question, that is ECG. ECG you have to learn very well. First, before learning the ECG, you have to first learn well about the cardiac cycle. Then only you can understand ECG. Okay. So first you have to study the cardiac cycle in detail, and after that reading ECG will make the things easier for you. Okay. So usually they are asking to draw the ECG and different parts. You have to label different parts of the ECG and what are the causes of different waves and intervals in ECG. Okay. So this is the normal ECG. We have the PR segment, QRS complex, S wave, etc. Okay. So in normal ECG, it consists of different waves, complex, intervals, and segments. And usually we have limb 1, limb 2, limb 3. Okay, there are 3 limbs. Usually the waves of ECG recorded by the lead 3 limb is considered as the typical waves. Okay. And the normal ECG, they are having the following waves, that is P wave, QRS complex, and T wave. So the normal is recorded from the limb 2, that is from the right hand and to left leg. Okay. Lead connecting the right hand and left leg is called lead 2, and that is usually considered as the typical waves of ECG. So this is the figure showing the normal ECG picture from the limb 2. Now coming to the major complexes in ECG. First is the P wave, and we are having the QRS complex or the initial ventricular complex. Third is the T wave or the final ventricular complex. And fourth is the QRST, the ventricular complex. We know that there are atrias, there are ventricles, there is interventricular septum and different SA node, AV node and things like that. Okay, so different parts are responsible for the generation of this major complexes in ECG. And first wave is the P wave. P wave is the positive wave. P for positive. Positive wave and it's the first wave in the ECG. It is called atrial complex. Why it is called atrial complex? It is because it is derived from the atrial region. And what is the cause of P wave? Usually, P wave is produced due to the depolarization of the atrial musculature. We have learned about the action potential. What is an action potential? Initially, there is a wave of depolarization followed by repolarization and then the resting state. Okay, that's the parts of the action potential. So, usually this P wave, QRS wave, complex, everything are produced by the depolarization. So, P wave is produced due to the depolarization of atrial musculature. We know that initial thing is the atrial contraction and P wave is produced due to the depolarization of the atrial musculature. Okay. And so we can remember like this ADEP, that is atrial depolarization, ADP, ADEP, that is atrial depolarization causes P wave. That you can remember like this. This question is repeatedly asked in multiple entrances also. Okay. So atrial depolarization is the cause of production of P wave. And atrial repolarization is not recorded as a separate wave. Okay. It is recorded along with, it is merging with the ventricular depolarization and hence atrial repolarization is not recorded as a separate wave. It is part of the QRS complex. We are measuring it as part of QRS complex or the second complex. Next is the QRS complex. It is also called initial ventricular complex. And here this is composed of different parts. First is the Q wave. The Q wave is a negative, a small negative wave. Okay. And it is continued as the tall R wave, which is a, so initially Q wave is a negative wave and after that there is a tall R wave and R wave is followed by a small negative wave, S wave. So initially a negative wave, then a positive one and after that a small negative wave. That comprises the QRS complex. Then coming to the what is the cause of QRS complex? It is due to the depolarization of the. So P wave and the QRS complex both are due to the depolarization. And P wave is due to the depolarization of the atrial musculature, whereas QRS complex is due to the depolarization of the ventricular musculature. And Q wave, it is due to the depolarization of the basal portion of the interventricular, that's the upper portion of the interventricular septum. The depolarization occurs that will be leading to the small negative wave, that is the Q wave. Next is the R wave, that is due to the depolarization of the apical portion of the interventricular septum and also the apical portion of the ventricular muscle. Next is the small negative wave, that is the S wave. It is due to the depolarization of the basal portion of the ventricular muscle. It is near the atrioventricular ring. So these are also due to depolarization of different parts of the, mainly the ventricular musculature of the heart. Next is the, so this is the QRS complex. We know that Q wave is a small negative wave. After that, a tall R wave. And after that, small negative, that is the S wave. This is a total QRS complex. Next is the T wave. And T wave is the final ventricular complex and it's also a positive wave. So T wave is a positive. P and T are positive. Also R is also a positive wave. And Q and S are negative waves. And now coming to the cause of T wave. T wave is due to the repolarization of the ventricular musculature. So you have to remember one thing. P wave is due to the depolarization. QRS complex is due to the depolarization. But T wave is due to the repolarization of the ventricular musculature. That you have to remember. Next is U wave. It is not always seen. It's an insignificant wave in the ECG. And it is also due to the repolarization. So T wave and U wave is due to repolarization, whereas P and QRS complex are due to depolarization. So U wave is due to the repolarization of the papillary muscle and the Purkinje fibers. Now coming to the intervals and segments of ECG. First is the PR interval. And the PR interval, it is between the onset of the P wave and onset of Q wave. Okay, that is the PR intervals. Between the onset of P wave and onset of Q wave is the PR interval. And it signifies the atrial depolarization and conduction of the impulses via the AV node. And it shows the duration of conduction of impulses from the SA node. First is the SA node, and it shows this PR interval, it shows the duration of conduction of impulses from the SA node to the ventricles via the atrial muscle and the AV node. And P wave, it represents the atrial depolarization. And usually short isoelectric or zero voltage period after the end of P wave, it represents a time taken for the passage of depolarization within the AV node. Okay. So the isoelectric period after the P wave, it represents a time taken for the depolarization within the, for the passage of depolarization within the AV node. Next is the QT interval. These things are having their clinical implications also. And QT interval is the interval between the onset of Q wave and the end of T wave. QT interval, it indicates the ventricular depolarization and ventricular repolarization, i.e. it significates the electrical. So electrical activity in ventricles, that is signified by the QT interval. QT interval means it represents the electrical activity in the ventricles. Next is the ST segment. It is the time interval between the end of S wave and onset of the T wave. It is also an isoelectric period. Okay. And next is the J point. It's a point where the ST segment starts is called. It's a part of the ST segment. The point at which where the ST segment starts is called the J point. And it's the junction between the QRS complex and the ST segment. Next is the RR interval. It's a time interval between two consecutive R waves. Usually this RR interval, it signifies the duration of one cardiac cycle. Now, this is also a very important question. The neuromuscular transmission. And what are the steps involved in the transmission of impulses across the neuromuscular junction in skeletal muscle with a flow chart? Okay. So, what is a neuromuscular junction? As the name indicates, it's a junction between the nerve terminal and the muscle end plate. Okay. So, it is defined as the transfer of information. So, neuromuscular junction is that it's a junction between the nerve terminal and the muscle end plate. And what is a neuromuscular transmission? It is defined as a transfer of information from a motor nerve ending to the muscle fiber via the neuromuscular junction. And it's a mechanism by which the motor nerve impulses initiate a muscle contraction. So the initial events that are leading to the muscle contraction are part of the neuromuscular transmission. So what are the events in neuromuscular transmission? First thing is that we have to release, when an impulse is occurring, there is a release of acetylcholine from the presynaptic membrane. Presynaptic membrane is a part of the nerve terminal and postsynaptic part is part of muscle end plate. So presynaptic is a part of neuron, whereas postsynaptic membrane, it is a part of muscle end plate. Okay. So initial step is that we have to release the acetylcholine. So we have, for example, muscles of our hand. We are touching a hot object. What happens is muscle contraction or the withdrawal of the hand is taking place. For that, the signal is transduced, i.e. the receptor is taking a particular signal, mechanical signal. It is transduced to electrical signals, that is activating the muscular contraction. So when any stimulus is received from the receptor, what happens? Received to the muscle, so it is carried away by the nerve. What happens in the nerve? In the nerve terminal, there are chemical signals that are produced by the neurotransmitters. Here, usually in nerve terminal relating to the muscle end plate, the transmitter is usually acetylcholine. There are vesicles containing acetylcholine inside the nerve terminal. So, initial step is the release of acetylcholine. It is due to the action of influx of calcium into the nerve terminal from the extracellular fluid. The calcium will be extracellular calcium will be entering into the nerve terminal, that is leading to the release of acetylcholine by the breakage of the acetylcholine containing vesicles. And this acetylcholine is secreted into the synapse or neuromuscular junction where its action is going to take place. So the first step is the release of acetylcholine. When the action potential it reaches the axon terminal, it opens the voltage gated calcium channels in the. So axon terminals, they are having voltage gated calcium channels. Okay. So this voltage gated calcium channels will be opening and it is causing the vesicles containing acetylcholine to release into the synaptic junction. So calcium ions from the extracellular fluid, it enters the axon terminal. So what is the structure that is responsible for the release of calcium? It's from the sarcoplasmic reticulum that is present in the muscle. So the calcium from the extracellular fluid that is entering the axon terminal, and this is causing the bursting of the vesicles by forcing the synaptic vesicles to move and fuse with the presynaptic membrane. We know that presynaptic membrane is a part of the nerve terminal. And now acetylcholine is released from the ruptured vesicles, and by means of exocytosis, this acetylcholine, it is releasing the acetylcholine. It diffuses through the presynaptic membrane and enters the synaptic cleft. Okay. So, you have to know the structure of this neuromuscular junction. This is the nerve terminal or the axon. These are the mitochondria for the secretion of this, making of this neurotransmitter. And we have the synaptic vesicles which are containing the acetylcholine. And after the entry of calcium into the nerve terminal, what happens is it will cause the vesicles to fuse with the presynaptic membrane and release its contents into the synaptic cleft. This is the synaptic cleft. Between the muscle end plate and the nerve terminal, we have the synaptic cleft, and the acetylcholine is released into the synaptic cleft. And the postsynaptic membrane, it's a part of the muscle end plate. Okay. So this part, it's called postsynaptic membrane. It's a part of the muscle. It is thrown into numerous folds. Okay. And they are making subneural cleft. So this is the structure of the neuromuscular junction. So after the first step, the acetylcholine is released into the synaptic cleft. Next is the action of the acetylcholine. So what's the action of acetylcholine? It's a chemical signal. Okay, this chemical signal is converted to electric signal in order to pass the information. This chemical signal has to be converted to electrical signal in order to get transmitted via the neurons. This chemical signal has to be changed to the electrical signal. Okay, after entering the synaptic cleft, the acetylcholine molecule, it binds with the nicotinic receptors that are present in the postsynaptic membrane. That's part of the muscle terminal. Okay, sorry, muscle end plate. And it usually this acetylcholine, we know that cholinergic receptors are of two types that you will learn in the second year. Two types, muscarinic and nicotinic. And nicotinic receptors are seen in association with the muscles. Okay, that you have to know. And so after entering the synaptic cleft, this acetylcholine molecules, it bind with the nicotinic receptors present in the postsynaptic membrane. And it forms, this acetylcholine is binding with the receptor and it forms a complex containing acetylcholine receptor complex. And it increases the permeability of the postsynaptic membrane for sodium by opening the ligand-gated sodium channels. So, after the binding of the acetylcholine with the receptors, what happens is ligand-gated sodium channels will be opening. We know that inside the cell there is a negative potential and outside there is a positive potential. Okay, that we all know. So after the release of acetylcholine, what happens is ligand-gated. Ligand means it is something related to some chemical transmitter or chemical signal. Okay, when a chemical molecule is binding with ligand-gated channels, this ligand-gated channels will open. So ligand-gated sodium channels will be opening and sodium from the outside of the cell will be entering inside the cell. So sodium is basically a positively charged molecule and inside the cell there is electronegative, that is minus 90 is usually the resting membrane potential. So when the positive charge is entering the cell, the negative potential is negative potential is reducing and it is coming more towards the zero. Okay, when adequate amount of sodium is entering the cell, the negative potential of inside the cell is reduced and it is raising to the zero. Okay, then what happens is, okay, and the sodium channels alter the resting membrane potential. It is initially 90, it will be coming to minus 70, like that. Okay, minus is reducing and develop the electrical potential called N plate. So initially a small amount of potential is released, that is called N plate potential. It will be small. When it is adequate, this amount of acetylcholine released is adequate, the adequate amount of ligand-gated channels will be opening and this N plate potential will reach the threshold and it will initiate the action potential. So next step is the development of the N plate potential. So N plate potential is the change in the resting membrane potential when the impulse reaches the neuromuscular junction. I.e., when acetylcholine is released, it binds with the receptor. It will cause the ligand-gated sodium channels to open and the sodium will enter inside the cell and it will raise the resting membrane potential. Initially, it is minus 90. When the sodium enters inside the cell, slight depolarization occurs. It may be coming to minus 60, which is called N plate potentials. It's minus 60, not 60. So that is called N plate potential. Multiple N plate potentials will together make it reach the threshold potential and then the action potential take place. So this we have already discussed. This is the structure of the neuromuscular junction. So what is the properties of N plate potential? It's a graded potential. It's not a single action potential. Multiple N plate potentials together makes the threshold potential and which will start the action potential. So N plate potential is non-propagative. It's not propagating. It is occurring only in that particular area. But action potential is propagative. It is traveling throughout the membrane. Okay. So, but N plate potential is non-propagative. It is occurring only at a particular portion that is activated by the acetylcholine. But it is leading to the development of action potential by making the threshold potential. So, next is the development of miniature N plate potential. Miniature N plate potential is a weak N plate potential in the neuromuscular junction that is developed by the release of small quantity of acetylcholine from the axon terminal. Okay. So it cannot directly lead to the development of action potential. And each quantum of this neurotransmitter produces a weak miniature N plate potential. And this amplitude of this miniature N plate potential is only 0.5 millivolt. That will not be making any significant change. And miniature N plate potential cannot produce action potential. When more and more amount of neurotransmitters released continuously, this miniature N plate potentials are added together and finally produce the N plate potential, and that will be resulting in the production of threshold potential and finally leading to the action potential. I.e., numerous multiple N plate potentials will be adding together to form the N plate potential, and numerous N plate potential together forms the threshold potential, and that will be leading to the action potential inside the muscle. When it is not leading to the threshold potential, then action potential will not be taking place. Okay. So this is the flow chart. Here, initially in the motor nerve fiber, an action potential is raised. This will lead to opening of voltage-gated calcium channels, thereby causing the entry of calcium ions from the ECF into the nerve terminal. This leads to the opening of vesicles. These vesicles will fuse with the presynaptic membrane and it will cause the release of acetylcholine. And acetylcholine is released to the synaptic cleft, and after that, the passage of acetylcholine will take place, and acetylcholine it will be binding with the nicotinic receptor that is present in the muscular end plate, and it will form the acetylcholine receptor complex, leading to the opening of ligand-gated sodium channels. Okay, and entry of sodium ions from the ECF. That will lead to another thing, that is entry of sodium ions from the ECF. Initially, we have the ligand-gated sodium channels that is opening, and after the entry of sodium ions into the cell, voltage-gated sodium channels are also activated, and multiple sodium ion is entering the cell. That is leading to the production of action potential. So here will entry of sodium ions from the extracellular fluid that will cause the development of N plate potential, and multiple N plate potentials together. Different muscle fibers, it together constitutes produce the threshold potential and leading to the generation of action potential. and excitation contraction coupling in the muscle occurs, and that will lead to the muscular contraction. Okay. So these are the basic sequence of event occurring during the neuromuscular transmission. This can also be asked as a short question. Next is after the production of neurotransmitters are very potent substance. So its action should not take place too long. Okay, its action is only about in milliseconds, and within that span of milliseconds, it can do its function. Okay, so destruction of acetylcholine, it can take place in two means. One is by the reuptake, one is by the destruction. Okay, so in this acetylcholine that is released into the synaptic cleft, it is destroyed very quickly, that is within milliseconds time, by enzyme acetylcholine esterase. There is an enzyme, particular enzyme called acetylcholine esterase, which is degrading the acetylcholine. The acetylcholine has to be degraded within a small span of time in order to prevent the exaggerated contraction of the muscle. Okay, muscle should not be contracted for too long. So in order to do that, we have to destroy the acetylcholine that is present in the synaptic cleft after its action has taken place. So acetylcholine is very potent, and even within a period of small milliseconds time, it's able to make its action. So rapid destruction of acetylcholine has some important functional significance. So what is the functional significance? It prevents the repeated excitation of the muscle fiber and allows the muscle to relax. Okay, exaggerated contraction can lead to hypothermia, etc. Okay, prolonged muscular contraction should not take place in the body, and hence this acetylcholine after its action is destroyed by different means. First is the reuptake process. It's a very precious molecule, so some amount of it is reabsorbed. Okay, so reuptake is a process that is occurring in the neuromuscular junction by which the degraded product of the neurotransmitter, it re-enters the presynaptic axon terminal where it can be reused. With the action of this acetylcholine esterase, what happens is acetylcholine is degraded into inactive choline and acetate. And choline is again taken back into the axon terminal from the synaptic cleft by means of a reuptake mechanism. There are many drugs acting in the reuptake sites. Okay, that you will be learning in the second year pharmacology classes. And there it is reused in the synaptic vesicle to form new acetylcholine molecules. So that's about the destruction. Final step is the destruction of the acetylcholine. So this is having 5 steps are there. First is the release of acetylcholine. Next is the action of the acetylcholine, development of end plate potential. After that, development of miniature end plate potential. And finally, after its function, this acetylcholine is destroyed. Okay. Next is describe the oxygen hemoglobin dissociation curve using a diagram, and list the factors which cause shift of oxygen dissociation hemoglobin dissociation curve to the right, and also add a note on hypoxia. This is also a very important question. So coming to the oxygen hemoglobin dissociation curve. So what is an oxygen hemoglobin dissociation curve? So it's a curve that demonstrates the relationship between the partial pressure of oxygen and the percentage saturation of the hemoglobin with oxygen. Okay, so it's a relationship. This curve shows the relationship between the partial pressure of the oxygen that is available and the percentage saturation of hemoglobin with oxygen. Okay, so what is the, it basically denotes the affinity of hemoglobin towards oxygen. So normally in blood, usually the hemoglobin is not saturated to 100%. It's only 95% of the hemoglobin is saturated with oxygen. And it usually depends on the partial pressure of oxygen. Okay. If the partial pressure of oxygen is more, then what happens is hemoglobin is having more affinity towards the oxygen. It accepts oxygen, and the partial pressure of oxygen is less, hemoglobin releases oxygen. I.e., in case of lungs, we know that in lungs, we have adequate amount of oxygen. Okay. So hemoglobin accepts oxygen from the lungs. Whereas in tissues, the amount of oxygen saturation is less, and hence hemoglobin will release its oxygen to the tissues. That will ensure the smooth functioning and distribution of the respiratory gases throughout the body. Okay. So if the partial pressure of oxygen is high, then affinity of hemoglobin towards oxygen will be high. Hemoglobin is unwilling to part its oxygen. Okay. When the partial pressure of oxygen is less, hemoglobin will be readily giving the oxygen to the tissues. Okay. So, what is a normal oxygen hemoglobin dissociation curve? Under normal conditions, it is S-shaped, okay, or sigmoid shape. And lower part of the curve indicates the dissociation of oxygen from hemoglobin, and the upper part of the curve, it indicates the uptake of oxygen by the hemoglobin depending upon the partial pressure of oxygen. So lower part indicates dissociation and upper part indicates the uptake of oxygen. It is all depending upon the partial pressure of the oxygen available. What is P50? P50 means it's the oxygen tension or the partial pressure of the oxygen at which the hemoglobin is saturated with 50% oxygen, i.e. the 50% of hemoglobin is saturated at particular partial pressure of oxygen. That particular partial pressure of oxygen is called P50. That differs in different conditions. Okay, normal P50 is usually 25 to 27 millimeter of mercury. When the partial pressure of oxygen is when the partial pressure of oxygen is about 25 to 27 millimeter of mercury, the hemoglobin saturation is about 50%. I blood contains 50% of the oxygen. At 40 mm of mercury, the partial pressure of oxygen, the saturation is about 75%. And it becomes 98% when the partial pressure of oxygen is 100 millimeters of mercury, then the saturation becomes about 98%. Normally, 95 is the maximum saturation level of the hemoglobin. So, around theoretical values. So, this is the oxygen hemoglobin dissociation curve. Okay. This indicates the dissociation and this indicates the okay. So, increase in P50 indicates the decreased affinity for oxygen, i.e. when more amount of oxygen is needed to saturate the hemoglobin to 50%, that means the affinity of hemoglobin towards oxygen is reduced. That will lead to the dissociation curve is shifted to right. So, when this curve is shifted to right, that means affinity of hemoglobin to oxygen is reduced, i.e. here this axis, x-axis indicates a partial pressure of oxygen, okay, and this indicates the percentage saturation of the oxygen. If it is shifting to right means more amount of, even in the more amount of partial pressure of oxygen is available, the saturation is only less, okay. That is shifting to right means affinity of hemoglobin to oxygen is reduced, i.e. Hemoglobin is willing to part its oxygen. Whereas shift to left means hemoglobin is tightly bound to oxygen. It is unwilling to give its oxygen to different tissues. That is shift to left. Okay. This shift to right indicates less affinity and shift to left means more affinity of hemoglobin. Okay. So, increase P50, it indicates a decreased affinity of oxygen and oxygen hemoglobin curve is shifted to right. And decrease P50, it indicates the increased affinity of oxygen and oxygen hemoglobin dissociation curve is shifted to left. And these are the main factors, coming to the different factors which is affecting the oxygen hemoglobin dissociation curve. In oxygen hemoglobin dissociation curve is shifted to left or right by different factors. Shift to left means acceptance of oxygen by hemoglobin. And shift to right indicates dissociation. Okay, that is shift to left means association or more affinity. Right means dissociation or less affinity. So, shift to right should be occurring in tissues normally. So, oxygen hemoglobin dissociation curve is shifted to right in different conditions like decrease in partial pressure for, i.e. normally tissues, there will be decreased partial pressure. So, hemoglobin will be giving more oxygen to the tissues. And increase in partial pressure of carbon dioxide, i.e. when the carbon dioxide concentration is high, then also oxygen hemoglobin curve should be shifting to right, i.e. affinity of hemoglobin to oxygen will be reduced. Increase in hydrogen ion concentration, that's the other effect for increase in partial pressure of carbon dioxide, and decrease in pH or acidity, that is also causing shift to right of the oxygen hemoglobin dissociation curve. Next is increased body temperature also shifts the ODC to right. Also, excess of 2,3-DPG. This also causes hemoglobin to lose its affinity towards oxygen and leads to more readily availability of the oxygen from the hemoglobin. It's otherwise called 2,3-DPG or BPG. Okay. Diphosphor or biphosphoglycerate. It's a part of, byproduct of EMP pathway of carbohydrate mechanism. and it usually combines with the beta chains of hemoglobin to make it more taut form, thereby releasing the amount of oxygen available in the hemoglobin. And in conditions like muscular exercise and high altitude, what happens is DPG will be increased and oxygen will be readily available to the tissues due to the less affinity of hemoglobin to the oxygen. And ODC will be shifting to the right. Next is shift to left. That means affinity of hemoglobin to the oxygen is more. In fetal blood, we know that fetal hemoglobin has got more affinity to oxygen in order to survive. Okay, it has to absorb more oxygen from the maternal blood, and hence fetal hemoglobin is having more affinity towards the oxygen when compared to the adult hemoglobin. And decrease in hydrogen concentration means decrease in carbon dioxide content and increase in pH or alkalinity. That is, this all means decrease in carbon dioxide content or increase partial pressure of the oxygen. So that will be leading to shift to left of the oxygen hemoglobin dissociation curve. Next is hypoxia. Hypoxia, we know that as the term indicates, hypo means something less. So it is defined as a reduced availability of oxygen to the tissues. Term anoxia refers to absence of oxygen, but absence of oxygen is not practically possible in a living body. Absence of oxygen is not practically possible. So hypoxia is nowadays used as a common term. Okay. So what are the causes of hypoxia? There are 4 important factors which can cause hypoxia. One is the oxygen tension in the arterial blood. When oxygen tension in the arterial blood is reduced, it will lead to hypoxia. Also, the oxygen carrying capacity of the blood. When anemia and things like that happen, what happens is the oxygen carrying capacity of the blood is reduced, thereby leading to hypoxia. Next is rate of blood flow. When the rate of blood flow to our different organs are affected, usually it occurs in the extremities. When diabetes and things like diseases like that, it can lead to reduced rate of blood flow to the extremities, etc., thereby leading to hypoxic damage, hypoxia, ischemia, etc. And utilization of some cells, they require, that is, for example, brain cells, they require more amount of oxygen for its survival. So according to different cells, the amount of oxygen needed for their normal functioning is different. So that can lead to hypoxia. The amount of oxygen needed for heart cell is different from brain cell, like that. So different cells have different oxygen requirement. According to that, hypoxia can occur to different. Initial, when the organs which are in need of large amount of oxygen will be initially affected in case of a hypoxic event. So there are different types of hypoxia. On the basis of different causes, it can be divided into 4 types. First is the hypoxic hypoxia. Second is the anemic. These are different stages. Okay. According to the different stages of hypoxia, cause of hypoxia. First is hypoxic hypoxia. Second is anemic hypoxia. Third is stagnant hypoxia. Fourth is the tissue toxic hypoxia. First is hypoxic hypoxia. Hypoxic hypoxia, as the term indicates, the oxygen available is less. It can be due to either the oxygen content in the blood as such is reduced. That is called arterial hypoxia. Or it is usually characterized by that is the initially. Even the amount of oxygen available is less. And rest of the features are normal in case of hypoxic hypoxia. The amount of oxygen available for the intake is reduced in hypoxic hypoxia. Next is, so what are the causes of hypoxic hypoxia? Low oxygen tension in even in atmospheric air, when the carbon dioxide or in carbon monoxide poisoning, what happens is the partial pressure of oxygen available is less. In such cases, the enough oxygen is not available, that is leading to the hypoxic hypoxia. Or it can be due to respiratory disorders associated with decreased pulmonary ventilation, which does not allow the intake of enough oxygen, right? Proper arterial oxygen content cannot be maintained in hypoxic hypoxia. Also, it inadequate oxygenation of lungs, which does not allow the diffusion of enough oxygen. Also, in cardiac disorders, in which enough blood is not pumped to transport oxygen. In this all cases, it can lead to hypoxic hypoxia. Next is anemic hypoxia. Anemic hypoxia, the amount of hemoglobin or the RBCs available for the carrying of oxygen to different tissues is less. So it's a type of hypoxia caused by the anemic conditions. And oxygen availability, here oxygen availability is normal, but the blood is not able to take up the sufficient amount of oxygen due to anemia. And anemic hypoxia, it is characterized by the decreased oxygen carrying capacity of the blood. So oxygen is available. Unlike the hypoxic hypoxia, oxygen is available, but the oxygen carrying capacity of the body is impaired. It can any condition that is causing anemia can cause anemic hypoxia. So it is caused by the decreased number. Either it can be due to decreased number of RBCs will be less, or decreased hemoglobin content, or formation of some altered hemoglobin. Okay. In certain conditions, altered hemoglobin, like in thalassemia, we can have altered hemoglobin. Okay. Hemoglobin structure is altered. And combination of hemoglobin with gases other than oxygen and carbon dioxide. Even carbon monoxide poisoning, what happens is carbon monoxide, hemoglobin is having more affinity towards the carbon monoxide than to oxygen. Okay, that can also lead to combination of hemoglobin with other gases. So the oxygen carrying cells are impaired in anemic hypoxia. The next is stagnant hypoxia. It is caused by the decreased velocity of blood flow. So it is called hypokinetic. Blood is stagnant, or the velocity of blood flow is reduced, and it is rest other features will be remaining normal in stagnant hypoxia. Just the velocity of blood flow is reduced. And what are the causes of stagnant hypoxia? It can be due to congestive cardiac failure, i.e. the pumping activity of the heart is impaired, and hence the blood flow, velocity of the blood flow will be reduced, and that is leading to the stagnant hypoxia. Can be due to hemorrhage, surgical shock, vasospasm, thrombosis, embolism. These are all causes for stagnant hypoxia. Next is the histotoxic. Here the oxygen availability is normal, oxygen carrying capacity is normal, but tissues cannot utilize oxygen. Even the oxygen is reaching the tissues, but tissues cannot utilize the oxygen. Why? Because of the impairment in the enzymes which are leading to the utilization of oxygen in tissues. So histotoxic means, histo means something related to tissue. Histotoxic hypoxia, it's a type of hypoxia produced by the inability of the tissues to utilize oxygen. Oxygen is delivered and all other features are normal, but the tissue and or the enzymes which are required for the utilization of oxygen is impaired in case of histotoxic hypoxia. So what are the causes? Cyanide or sulfate poisoning. Here some enzymes are degraded which can lead to histotoxic hypoxia. So these substances can degrade the cellular oxidative enzymes, and there is complete paralysis of the cytochrome oxidase system. So even the oxygen is supplied, the tissues are not in a position to utilize it. So this is a table indicating different. So here in hypoxic hypoxia, only the partial pressure of oxygen in arterial pressure is reduced. Rest of the things are normal. Oxygen carrying capacity, everything is normal in hypoxic hypoxia. Whereas in anemic hypoxia, even the partial pressure of oxygen is normal, but the oxygen carrying capacity is reduced in anemic hypoxia. Rest of the things are normal. In hypoxic hypoxia, 100% oxygen therapy. Efficacy for when we treat with the oxygen, we are giving adequate amount of oxygen, this hypoxic hypoxia can be cleared. In anemic, when we give oxygen, some amount of the effects can be corrected. 75% of the effects can be corrected by giving the oxygen therapy. Stagnant hypoxia, only less than 50% is corrected by means of oxygen therapy. Okay, here the velocity of the blood flow only is reduced. Histotoxic, the tissues are impaired, and hence oxygen therapy is not useful in case of histotoxic hypoxia. The tissues itself are damaged. Next is reflex arc. So reflex means it's a rapid involuntary movement that is required for the prevention of further tissue damage. When a destructive stimulus, in case of say a needle prick, etc., there is a starting of reflex arc. Such reflexes are to be rapid, and hence it has to be bypassed. It has to bypass the brain, and it should travel through the spinal cord. Okay, so this reflex has to be very rapid, and usually this reflex arc is occurring between the effector organ and the spinal cord. So it has got 5 components basically. Reflex arc is having 5 components. First is the receptor. So receptor is a site of injury in case of, for example, when a needle prick occurs, the site, that needle prick is carried by a receptor. We are touching a needle with our finger, and there is a receptor which transduces that mechanical stimulus to a chemical stimulus. Then only it can be transmitted to the spinal cord and appropriate action can be taken. Okay. So receptor, it's the end organ which is receiving the stimulus. So first portion of the receptor arc is, reflex arc is the receptor. So it's an end organ which receives the stimulus. That's the mechanical stimulus is received by the receptor and it is transducing that mechanical stimulus to electrical stimulus. And when the receptor is stimulated, impulses, that is electrical stimuluses are generated in the afferent nerve. So the second part is the afferent nerve. So stimulus from the receptor is carried to the spinal cord by, usually this reflex arc bypasses the brain because in order to do the action very rapid, in a rapid manner, we have to bypass brain. And this reflex arc is taking between the spinal cord and the end organ. Okay. So afferent nerve is the second component of the reflex arc. And the afferent nerve or the sensory nerve, it transmits the sensory impulses from the receptor to the center. Usually it is the spinal cord. It's the ventral horn of the spinal cord. Motor reflexes are carried by the ventral horn of spinal cord. So this afferent nerve or the sensory nerve transmits the sensory impulses from the receptor to the center or the spinal cord. Next is the, so first is the receptor, second is the afferent nerve, third is the center. So center receives the sensory impulses via the afferent nerve and it in turn, it generates the appropriate motor impulses are generated in the center. And usually the center is located either in the spinal cord. In reflex arc, in most cases, it is located in the spinal cord. In simple reflex arc, the synapse between the, in simple reflex arc, sometimes the afferent nerve and efferent nerve are connected by interneuron or internuntial neurons. Then they will become the center. Interneurons will become the center in simple reflex arc. Usually, sometimes the impulses are going to the spinal cord, usually the ventral horn, that acts as the center. Next is the, so reflex is carried from the receptor via the afferent nerve to the center, and from the center, the information, the response is taken away by the efferent nerve. Efferent nerve is coming to the efferent effector organ. Okay, so efferent or the motor nerve, it transmits the motor impulse from the center to the effector organ. So next part is the effector organ. So first is the receptor, second is the afferent nerve, third is the center, next is the efferent nerve and effector organ. And this impulses from the center is carried by the efferent nerve towards the effector organ. So it is usually, these impulses are reflex are constitute mainly muscle or gland. Okay. So this motor response is carried via the efferent nerve to the effector organ where the activity occurs in response to stimulus. And afferent and efferent nerves, some in simple reflex arc, they can be connected by interneurons or internuntial neurons. In such cases, this internuntial or interneurons, they can also act as a center. Okay, such neurons, they are also called internuntial neurons, interneurons or connector neurons. So these are the 5 parts that are involved in the reflex arc. This is also multiple repeatedly asked question in different university papers. Okay, so now coming to some important topics, I will tell you some important topics which you have to refresh before going to the exam. So this is a simple reflex arc. Here is a receptor. The stimulus is carried by the effector nerve towards the center. Here in simple reflex arc, we may be having a connector neuron or the internuntial neurons. Then it acts as the center. Usually it is spinal cord. And from there, the efferent nerve is coming. It is leading to the effector organ. Usually it's a muscle or a gland. Okay. So these are the important topics. Anemia you have to study. Deglutition, different stages. It is asked multiple times in different papers. Also coagulation of blood, intrinsic, extrinsic pathway, disorders of coagulation. These are also very important. Blood grouping, surfactant. That's an important short note. Pituitary, thyroid, adrenal cortex. These hormones are in the endocrine system. These are also repeatedly asked. In cardiac system, cardiovascular system, You have to learn well about the cardiac cycle first. That's a base. Then only you can learn other things. You can add on this cardiac cycle. Okay. What is cardiac output? What is the factors affecting the heart rate? Like that. Juxtaglomerular apparatus. This is also asked multiple times. Then muscle contraction. Sorry, it's contraceptives. In reproductive system, you have to learn about the contraceptives. Also, spermatogenesis also is asked in one of the papers. Contraceptives is very important in reproductive system. So these are the important topics you have to cover before the exam. Okay, you have to just go through these things. These are the last topics for the last minute revision. Okay, so thank you for your patient listening. Thank you. Good night.